SMC Consult, so much for Christ, located at Adenta. We are into medical advocacy and consultancy, science advocacy and consultancy, IT or information technology, digital marketing, and into publications. Trust us, count on us for all your works, for all your medical screenings, for all your scientific consultations, for all your ICT work and digital works, and also, lastly, for publications. SMC Consult, so much for Christ. SMC Consult, to the ends of the world.
Good afternoon. My name is Vanessa Isinabeki. I work at the EPP. We have different sections here. We have Zealand, we have
With me today is an astute academician who went to the University of Ghana and studied biochemistry both at undergrad level and at the master's level. Let's welcome to the show George Youngson.
Thank you, Solomon.
Welcome, George.
Yeah, thank you. I'm pleased to be here.
Nice to have you here.
Great.
So what have you been up to?
Okay. So I just completed my MPhil
In which
biochemistry, where I was looking for novel compounds in the treatment of breast cancer. And yeah, so that's what I've been doing. And after submitting my thesis, I've been around. I've been managing a clinic on part-time basis, and I also teach chemistry in private schools.
Wow. So do you love anything with chemistry in it?
Yes.
Wow. So who is George?
For George, what actually defines me is, I'll just say, honesty, hard work, and integrity. And this is what I carry on anywhere I find myself. So you see me, you send honesty, hard work, and integrity.
Honesty, hard work, and integrity. These are serious, true values that we have to uphold. Anyway, so where did George have his basic education?
So my basic education was at Redwana Comprehensive School.
Redwana?
Yes,
wow.
Redwana.
Where is it located?
At Zenu, at Adeka.
Atadeka, which region?
Somewhere, Greater Accra, somewhere around.
Wow, it's in Accra?
Yeah, Ashaman area.
Ashaman area.
Yes.
Wow. Shout out to Ashaman people. Okay, so secondary school, where did you go?
So I attended Presbyterian Boys Secondary School.
Wow, Legon.
Yeah, Legon.
Wow, so you're a Blue Magician, or that day?
Sure.
Wow, which year group?
So I completed 2012.
2012.
Yeah.
So when you were here in secondary school, what was your best subject?
Chemistry, of course.
Still chemistry?
Yes.
Wow, everything chemistry.
I just get it. It makes sense.
You really understood the mystery behind the kit.
Exactly.
Wow. That's nice.
Yeah.
Okay, so can you tell us more about your project, what you did at the master's level? Can you tell us more?
Okay, basically at the master's level, I was, so there was an in silico work. In silico work means using computer to actually determine the mechanism of action of a compound. So you do it, so it's a dry lab thing where you use computer to find compounds that, the mechanism of action of compounds. So that had been done already, and what I did was I was supposed to validate that in vitro. So yeah, before a compound will be accepted as a drug, a potent drug, it has to go through processes. So once you have done the in silico work, which is the dry lab thing, you have to validate it in vitro before we go to animal model, before we start clinical trials.
Wow.
So so a group in chemistry department did the in silico work. So then I had to validate the results in vitro, because the compound proves to be cytotoxic.
To my hearers or my listeners, what does it mean to say cytotoxic?
So cytotoxic means it has the ability to kill cells.
Yeah. living cells.
Yeah, living cells.
Okay,
Yeah.
including animals and every other thing.
So it is cytotoxic, but this compound was very cytotoxic to cancer cells.
To cancer cells.
As against normal cell lines.
So
that
your aim was to treat cancer?
Yes. So the target was to get a compound that we can use that has the potential for treatment of breast cancer. That was the target.
Okay.
So in doing this, what I did was I got the compound and I treated breast cancer cell lines with it. So breast cancer cell lines are actually cell lines that have been taken from a lady or a woman with a breast cancer.
Okay.
Then has been maybe normally they use laser to separate them into single cells.
Okay.
So this single cells is what we culture in the lab. Then after culturing it, we try to treat them with a compound of interest. Then we have certain factors we look at, which could be in terms of the sublethal concentration of the compound.
I think you have to break it down for us at this stage. So first of all, what do you mean by culture? Because the one we know is in social studies.
Okay, so when we say culturing cells, it's about growing the cells in profusion. Profusion means that you want to get more of the cells.
Okay.
Yeah, so what we do is we normally grow cells in a nutrient-rich medium. Okay. That's medium that has a lot of amino acid, glucose, and other minerals to help a normal cell to grow. So it's more like just as human beings we eat to gain nutrients. Cells, so that means that all our cells also need nutrients for growth. So so we just supply them directly the end product of digestion, more like amino acids, glucose, instead of going to take banku or something. They are cells, so we have to give them the end product of digestion.
Wow,
glucose.
To make it simple for them.
Exactly.
Okay, viewers, this show is being sponsored by SMC Consult, So Much for Christ, EPP Bookshop, Brainwork, and Aspa Drinking Water. Aspa Drinking Water is located at Denta, and EPP Bookshop is located on the Medina stretch of the Accra-Medina Road at Legon, precisely, at the University Mall. It's on the second floor of the high-rise building over there. Trust EPP for any kind of book that you want. Thank you. So back to you, George. How would you rate your research? How far in terms of impact on cancer cells, especially breast cancer?
Oh, okay. So the compound happens to prove potent against breast cancer.
Okay.
That is, it was able to kill the breast cancer at a very low dose.
Okay.
And also, it was able to inhibit the breast cancer from moving to other parts of the body.
Okay.
From the data I collected.
I think I know one word for spread of those things. Metastasis.
Exactly, that's metastasis.
Okay.
So that makes the compound more macrostatic.
Wow.
So, which means it will be able to slow down the movement of the compound.
Macrostatic.
Yeah.
Viewers, we've learned something new. Macrostatic.
Yeah.
Wow.
Yeah. So basically that was what my work is. I employed a lot of tools such as PCR, RTQ PCR. And
can you tell us what PCR means?
Oh, more like, so PCR is a technique, it's a tool we use to amplify a gene in the lab.
Okay.
Yes. So, but RTQ PCR is a modified version of PCR where we try to quantify the genes.
Okay.
Yeah.
Do you know the full name for these abbreviations?
Yeah, so RTQ PCR is reverse transcriptase quantitative PCR. That's polymerase chain reaction.
Oh, okay, nice. A mouthful of words. Reverse transcriptase quantitative polymerase chain reaction.
Exactly.
Interesting, nice one there.
Yeah.
So, looking at biochemistry and the world, especially taking Ghana into consideration, what are the good and bad sides, the up and down sides, the highs and the lows?
Concerning biochemistry.
Yeah.
So, for Ghana, I think the government has not been fair to biochemists, because as the year goes by, there are new biochemists coming into the system, and mind you, each of these people have done research.
Okay.
So I'm wondering, all this research, where do they go? Who picks the research and try to use it to solve national problem or maybe health issues? So it ends there. So my undergrad project like this, which was about something to do with fishes, where you have a pond in which the fishes were dying, you have one in which the fishes were surviving. Right after I was able to, make some findings where I was able to find that algal bloom was the cause. At least I had a defined protocol in identifying the cause of the fish death in the pond. After undergrad, the project is lying somewhere.
Wow.
So it ends there.
Sad story.
Yeah. So I'm thinking that going forward, the government could set up a data hub, sort of, such that both all our undergrad projects and all our undergrad findings actually goes there.
Okay.
And we have team of people that as year goes by, they are able to analyze the project and see that, okay, this one will solve this problem, this one will solve that problem, this one has a potential of solving this health issue locally.
Okay.
And maybe we can take it up for maybe a PhD or MPhil, what's the name? Project to validate those results, then we can move up from there. Because my undergrad project is just...
They are all lying there.
They are all lying
White
there.
elephants.
Exactly. And also, I hope the government can also sponsor some of it, because my MPhil was sponsored by WACBIP.
Oh, okay. What's WACBIP?
West African Center for Cell Biology of Infectious Pathogens.
Wow. Where is it located, please?
Oh, University of Ghana.
Oh, okay.
Yeah, the Biochemistry department.
Okay.
Yeah. And at least they sponsored my second year MPhil.
Oh,
yeah, they
nice
paid my
them.
MPhil.
Thank you, Agbe.
Yeah, they paid my MPhil fees and my second year MPhil fees. And they also gave me a project funding of $5,000 US.
Wow.
And I was getting a monthly stipend of $100.
Hey.
Yeah.
They are rich.
Yeah.
You have to come and pay tithes to me.
Yeah, I wasn't rich by then, but I think WACBIP was trying to, spark the interest of science in me by giving me all those packages. And I think they were really helpful because I had sound mind to do my research. And I know my findings are great, but just that the data are yet to be published.
Oh, okay.
And I hope when they come out, they'll be able to solve issues with breast cancer, because now Ghana, one of the most prevalent cancer in Ghana now is breast cancer. Yeah.
Okay. So before we tackle other issues, I want to ask you, what do you mean by publication? Can you tell us? Throw more light on publication.
So publication is like, once you get your findings, you have to, design your findings into an article.
Okay.
More like a newspaper, let's say a newspaper.
Okay.
But in the scientific world or any in academia, we call it a journal.
Okay.
So all your findings have to be, you send it, and when you send it to maybe a journal, they have to scrutinize it, go through it, and ensure that the information you are putting across are right and everything, and are novel.
Okay. What novel means?
Novel means new. Okay. Nobody has done that before. Okay. Or even if someone has done that, there is a build-up.
Okay, so not a direct copy?
No. So something different that you can add to knowledge, because the agenda here is adding to knowledge. So once the information is novel, they then publish it, and yeah.
Wow.
So that's how it goes.
Wow, that's nice.
Yeah.
So in your profile, you said you teach.
Yeah, I teach.
Where do you teach, please?
Okay, so I teach at ODADE College currently.
Oh, ODADE College. Behind Pacesetter.
Yeah, behind Pacesetter.
How can you feel?
Yes, how can you feel?
Wow, so they don't feel at all?
It's a marketing strategy, so
something
like that.
But I'm sure your students pass all the chemistry.
Well, yeah, they do.
Wow,
yeah.
Wow. So for the second cycle level, somebody who is looking forward to coming to the tertiary or the university, and the person wants to come and do biochemistry as a course, what are the factors or things he has to put into consideration?
So first, when I was in SHS, the whole agenda was to do medicine.
Okay.
So I was fighting for medicine, and when I didn't get medicine, because I actually had a grade for medicine, but I wasn't called for an interview, so then I just leaned back. I said, okay, if I'm good enough to do medicine, then I have to prove to people I'm good enough to do biochemistry. So that was the mindset. So when I went to the school, I was studying hard. At least I was rocking shoulders with some of the medical students in terms of their scores, physics. I was also getting all the good grades. So after the first year, the plan was I can reapply for medicine. But when I saw my results for the first year, they were great, so there was no need to go and start something new. So then I said, okay, let me just be here.
Wow.
So I did the biochemistry and I was successful. And it's not a course that you should like. When you are going the mindset with the mindset that it's an option. I didn't really like it, and I'm here.
Okay.
If you are good enough to do the medicine, prove to the world that you are good enough to do the medicine by learning your biochemistry. And that doesn't make biochemistry second to medicine.
Okay.
But in my nurse world, the campaign, not in my nurse world, currently the campaign I've been doing is to tell people to choose biochemistry as a first choice because it's very, very interesting. And it's something I work with. I understand it that much that I work with it. It's a concept and it's with you. It's interesting. It makes it very interesting. And with biochemistry, you are more like a handyman. When it comes to pharmacy, you can talk a little bit when it comes to toxicology. When it comes to medicine, you can at least talk about some biochemistry that maybe leads to pathogenesis and all those things. We are almost everywhere. When in the food industry, we are there in yogurt production.
Is that why people who do other professions sort of pick up on biochemists? Because you are like the all-round player.
Yes, so all you need is just trainers and we are there. We can,
you deliver.
Yeah. So we are termed as handymen.
Oh, okay. Handyman.
Yeah.
So you're a handyman.
I guess so. But now I'm a professional because now I have delved into a field which is cancer biology.
Okay.
Where I'm mainly into drug discovery.
Drug discovery.
Yeah.
You know, Pastor Mensa Otabo has a saying that scientists don't manufacture knowledge, they discover knowledge. Meaning the thing was there, but it was covered, and they took the cover off. So discovery must lead you to faith and devotion in God. So I think, George, you are doing a good job, and keep it up. We pray for more grace for you.
Sure.
Yeah. And I'll just ask another question. So right now, the students at the second cycle level, let's say those writing the WACC right now, what are the subject areas you need to pass to get maybe biochem?
Oh, your chemistry, your biology, your elective maths, and 3 other core. For me, I prefer aggregate 6.
Aggregate 6?
Yes, so from 6 to 8, you should get in.
Okay.
Yeah, from 6 to 8, you should get in. You are assured of getting in. Yeah.
George is a living testimony of no medical suicide.
Yeah.
You see, or suicide. People who have the notion of medical or suicide. If you don't get medical, then you're going to kill yourself. You write a note for your parents. Mommy and daddy, I'm sorry. I didn't get medical school. I'm sorry. That's something we don't want to encourage in the 21st century or 22nd century. Yeah. So how about the future job occupations available after reading biochemistry?
Okay, me like this, I want to be a pure research scientist.
Oh, okay.
Because I like the fun when it comes to research, the whole fact that things might not go as you expect, the fact that you try and give hypothesis, but at the end it doesn't go as expected. You sometimes meet it and you get excited. So I really love research.
Okay.
But when it comes to other job opportunities, like the careers, so you can work in the pharmaceutical industry where you'll be doing the work I started with.
Okay.
Yeah. And we have a couple of biochemists in Mampong.
Okay.
Yes, we have a couple of biochemists in Food and Drug Authority.
Okay.
We have a couple of biochemists in Standard Boards.
Okay.
You are just a handyman. You are all over the place. So we have biochemists in Nestle Ghana. We have biochemists in Unilever Ghana. We have biochemists in
I think Blue Skies.
Yeah, Blue Skies, what's the name? CSIR.
CSIR.
Yeah, so biochemists are all over.
All over the place.
Yeah, but the thing is about your interest and your placement.
Okay.
In Ghana, we don't have our way of thinking. If I don't go here, if I don't get here, then it ends there. No. Biochemistry, you are a handyman. Once you are trained, so I did biochemistry and I was managing a clinic.
Okay.
That's the kind of training, at least for biochemistry in University of Ghana you get.
Okay.
And you can work anywhere. So I was managing a clinic and I wasn't terrible. At least when I went there, they couldn't pay their workers. But at least when I went there, they started paying their workers on time. So yeah.
So does it mean you played a role in the managerial activities of?
Yeah. So
wow.
I was, if the in charge is not, I was the one there managing.
So
it.
you caused the payment to
so
flow.
at the end they started paying them on time. Initially they used to pay them the following month.
Isn't that your
first 2 weeks.
intrinsic skill that you had, or you are giving credit to University of Ghana for that?
Oh, I think I'll give that. I had it by University of Ghana really, because University of Ghana Biochemistry, you have an assignment, you have to submit it on time.
Okay.
There are things like Excel, your Word document, PowerPoint presentations. These are skills I didn't know before I entered biochemistry in University of Ghana. But when I completed, at least I'm able to know. And the course also makes you very analytical. So you don't have to go and do managerial course or anything to be able to get it. But at the end, you're able to, read in between the lines and make decisions that will benefit the company.
So would you agree to the assertion that it's a holistic training?
Perfect. That's the perfect word.
Okay, so George is telling us that the biochemistry department of University of Ghana has made him become analytical, and he has received holistic training. This is an impressive one from him, and I think it will give kudos to University of Ghana for such a good training. Was it tedious?
Yes, very.
And how was the duration, course duration?
Course duration.
How many years?
Oh, it's 4 years.
It's 4 years.
Biochemistry is 4 years.
Okay, University of Ghana. After which you can do an MPhil.
Yeah, MPhil. Yeah.
Are there branches in the MPhil?
Oh, so there is molecular biology of infectious diseases, and also I did purely MPhil in biochemistry. There is MPhil in molecular biology as well.
Okay.
Yeah. So for University of Ghana, those are the fields.
Wow.
Yeah.
Before we wrap up, I want you to give a shout out to some people.
Oh, shout out. So I'd like to say thank you to University of Ghana, specifically WACBEP. I think they've really sparked the interest of research in me, and I'm very, very excited for the platform they gave me. There are other things WACBEP have helped me with that are in the pipeline. I can't say it now. And also, I'd like to say thank you to all friends for their support. I'd like to say thank you to my family, my loved ones, and people who keep praying for me. And finally, to Presec.
Wow.
At least they gave me the start in science, and I'm so much excited to.
Will you sing the anthem for us?
Oh, of course.
Okay. Do us the honest.
Oh. Happy are we, studious are we, students of Presbyterian Secondary School. Onwards we march, we trudge along to happy victory, to victory, to victory. Our motto is a solid bulwark propping us along. Illuminate you with the beamers lumen. In the light, we shall see light. Illuminate you with the beamers lumen. For Christian training we get a sure solid foundation to take our places in the future of our country and church. For Christian training we get a sure solid foundation to take our places in the future of our country. Oh, come along and join us. Come along, join us. Happy studios are we. Wow, we thank you so much. You do one last thing for us, but before then we'll do a signage of Science Digest. When you say Science Digest, you say yam no feko. Sure.
So Science Digest.
Yam no feko.
Then lastly, you're going to dance for us.
Oh, that'll be serious.
Yeah,
really?
so we'll give you a song titled Jerusalema, and then you dance to it for us. Youngson, we are very grateful and delighted to have you on the show, but we'll not leave you to go just like that. EPP has got your back from biochemistry. From their shelves, they have a nice book for you, which is a must read for everybody. Mm-hmm. So our able secretary Grace will bring us the book so that we do the honor of presenting it. Okay. Thank you very much. Okay, so this is the book, Outlines of Biochemistry, 2010 African reprint by Eric E. Korn. A must read. on the shelves of EPP. You can get it at Legon on the second floor of the high-rise building on the University Mall. So George Youngson, we are grateful for everything, and this is our thank you to you from SMC Consult and Science Digest show. So George Youngson, we are grateful to have you on the show, and EPP has got your back with this book, and we are presenting it to you. But I would have loved to shake your hands, but for Corona's sake, Corona be hard drink.
Okay, thank you very
You're
much.
welcome.
So I'm pleased.
You're welcome.
Uhambe nami zungangishilana dawo yami ayikholana buso wami awukholana ilondoloze zuhambe nami dawo yami ayikholana.
Viewers, we're wrapping up the show, and thank you for sticking around. It's been Science Digest, Yayam Nefiko, sponsored by SMC Consult, EPP, Brainwork, and Aspire Drinking Water. Join us another time with an astute teacher from Presbyterian Boys Secondary School. See you there. Bye.
SMC Consult, so much for Christ, located at Adenta. We are into medical advocacy and consultancy, science advocacy and consultancy, IT or information technology, digital marketing, and into publication. Trust us, count on us for all your works, for all your medical screening, for all your scientific consultations, for all your ICT work and digital works, and also, lastly, for publications. SMC Consult, so much for Christ. SMC Consult, to the ends of the world.
Good afternoon. My name is Vanessa. I work at the EPP. We have different sections here. We have Zealand,